Hi, my name is Brandon Miles, and I'm here to explain our lab experiment of enzyme activity. And I first would like to talk about trypsin and just explain how that works. Trypsin is an enzyme that breaks down protein by the use of hydrolysis. It is also found in the small intestines of humans and animals. Trypsin digests protein in the diet. In our experiment, we wanted to figure out how much enzyme activity changes as pH increases. Our hypothesis was basically that enzyme activity will increase to the optimal point and will level out as pH increases. Enzyme activity was tested when trypsin was added to the centrifuge with the buffers and azo casein. Our results showed that pH, our results showed that there was a significant difference in absorbance between 6 and 8, pH of 6 and 8, and also 7 and 8. Where they were considered significant because it was 6 and 8 were 0.31 and 7 and 8 was 0.34. And those are both less than 0.5 and when 0.05. And when they're less than 0.05, they have a significant difference. But pH of in the pHs of P6, pH of 6 and 7 and 8 and 9, they were larger. They had a higher number than 0.05, and it was not significantly different. Our hypothesis was supported because enzyme activity increased until it reached the optimal point, which was proven in the differences between 6 and 8 and 7 and 8 were significant. So I would like to talk about enzymes. Enzymes are molecular catalysts that speeds up the chemical reaction. Many enzymes change shape when substrate binds. Enzymes are also very, very important in humans because without enzymes, we will not be able to live because the reactions will be too slow. When the reaction was catalyzed by trypsin, since trypsin is an enzyme, it sped up the reaction. So that's what trypsin did when we added it to the buffers and the azo casein. So as we saw the results that certain things were significant and others were not, we said that the decreasing of the pH will cause bonds/forces to break in the protein, and the shape was changed, and then which will alter the size and alter the size of the protein, and the protein will not be able to function correctly. The optimal pH will affect enzyme activity. For materials, we use 450 UL of reaction buffers, 500 UL azo casein, and we added trypsin to the centrifuge as well. We let this sit for about 20 minutes, and by sitting it for incubating it for 20 minutes, it allows the peptide bonds and amino acids to be released. Then right after that, we quickly added the 500 UL of 10% TCA, and that stops the reaction. After seeing this, we saw that there were two things, one that we did need and the other that we didn't. There was a pellet and a supernatant. The supernatant we needed, so we separated it and put it in a separate in a separate cuvette, and put the cuvette in a spectrometer to measure the absorbance and at 440 nanometers. Our experimental variables were pH 6, 7, 8, and 9, and they were all being tested of enzyme activity. Our control variables were put at 37 degrees Celsius, and a salt concentration of 0.15. The negative controls were used for negative controls were used for each of the pHs and that were being tested, but trypsin was not added to the negative control. We used water instead of trypsin for three replicates for our experimental variables or experimental group. So basically, there was 3 replicates in our negative control. Lastly, many groups did not had the same weren't the same. For example, Group B, they in their experiment, their p-values when comparing absorbance to 7 and 8, they were significant, while ours were not significant because their absorbance at 7 and 8 were above 0.05. So that means when it's above 0.05, it is significant. And I'm thinking this may have happened because the large variability being our variability, our sample for pH 8, or the difference of absorbance between the two set of data samples at the pH of 7. Thank you.